Today's lecture will be quite complicated, little bit difficult, little bit difficult to understand. So what I'm asking you, kindly pay me full attention today because it is quite an important one. We are talking about dilution and concentration of urine, how it is produced, right? So in a reference man, I told you long ago, what is a reference man? Is a man of 22 years, right? He is having 70 kilo body weight, living in a comfortable ambient temperature, and eating a diet of 2,500 to 2,800 calories. That is the reference man. Now, reference man, his body weight, 60 to 65% body weight is water, right? All human beings, they drink 2.5 liters to 3 liters of water in the form of drinking, 1500 ml, in the form of food and drinks, 1000 ml, and metabolic water, which is produced as a result of oxidation in our body. That is, take the example of carbohydrate oxidation, CO2, water, and energy, right? So this is how 300 ml of the water daily produced in our body loses water in the form of urine. That is, whatever the amount of water which we are drinking, usually we lose it in urine, 1500 ml, and then sweating. Now, sweating is, say, in case of winter, the sweating is minimum, therefore more water in urine, and in the summer, more sweating, therefore lesser water in the urine. So these two, say 1500 and 700, they are intermixing, depends upon the type of the weather. And then in the respiration, we lose nearly 300 ml of water in 24 hours. And then some kind of what we call as the insensible perspiration. That insensible perspiration always oozing out of our skin. This we do not notice it. When the sweating is appearing, we notice that water is coming out. But insensible perspiration, sweating is also perspiration. Insensible perspiration means water is coming out, but we don't understand it. We don't notice it. And then we lose nearly 100 ml of water on an average in the stool in the morning time. So thus daily intake of water output and input in ambient temperature is well balanced. What the kidney has got two important functions. That is, number one, that there are many functions, the two important functions we talk about. That it eliminates all the body waste, soluble body waste in the form of urine, and it prevents excessive loss of water also to maintain the fluid balance. So kidney excretes waste product and at the same time maintain body fluids by preventing excessive loss of fluid in the form of urine. Then comes the tubular fluid becomes concentrated, as you have seen, tubular fluid becomes concentrated as a result of water in solute reabsorption and excreted as urine. That is, here we are coming to an important point we will talk a little later. Normally, urine contains 95% water and 5% solutes. Now, most abundant solute right is urea. We excrete nearly 25 grams of urea every day. We are producing 25 to 30 grams of urea in the body every day as a result of metabolism, and that is excreted in the urine every day. That's the larger abundant solute. And then comes the second one comes the sodium chloride and other salts, creatinine and uric acid. These are the product of excretion in the urine. Please try to remember, in urine there is no glucose, no protein, no ketone bodies, no bile salt. That is, urine is free from these at least 4 substances. And then we come to, under physiological conditions, 180 liters of the glomerular filtrate is produced in 24 hours. Again, I am talking about the reference man. 124 hours, 180 liters. In case of female, it is 150 to 160 liters, right? and filtrate is produced in 24 hours. Out of 180 liters of the fluid, 178 liters of the filtrate is reabsorbed by the renal tubules. This occurs 60 to 70% in proximal contratubule reabsorption, 15 to 20% in the loop of Henle. Now try to remember, loop of Henle contains descending limb and ascending limb. So, and then 10 to 12% in early distal convoluted tubule, 5 to 10% in the late distal convoluted tubule and collecting ducts. Now, this reabsorption of water in late distal tubule and collecting ducts is under the influence of ADH, anti-diuretic hormone, which you are going to study very soon in posterior pituitary. Now, urine formation. Glomerular filtrate, which we have seen, say 180 liter, is produced every day. While passing through the renal tubule, it faces many changes. It changes in the volume, that is from 180 liters is reduced to 1.5 liter. 125 milliliter of glomerular filtrate is reduced to 1 mil. And that is the change in the volume. Composition totally changed. It contains glucose, it contains other things which are not present in the urine. They are completely reabsorbed. Urine is a product of concentrating and diluting abilities of the renal tubules. When the renal tubules have got ability to concentrate, absorb water and secrete some substances, and this is how the renal tubules are modifying the tubular fluid. All the cells in the body functions properly when they are bathing in extracellular fluid of proper osmolarity and concentration of electrolytes. Ladies and gentlemen, let's try to understand what is the osmolarity. Osmolarity refers to the osmotically active particle in a solution. Osmotically active particle, right? That is not the weight, but osmotically, number of the osmotically active particle in the solution is the osmolarity. The renal interstitium, again, is another term which we are using. Interstitium means some sort of ECF in the renal medulla. That is the interstitial space between the renal tubules and blood vessels. That is the ECF in the renal medulla is known as renal interstitium. Why do we give this special name? If we give, we can say that the ECF in the kidney, that is not sufficient. But I may say ECF means ECF everywhere. When we say renal interstitium means ECF in the renal interstitium, which varies in the composition, we will see just now. And therefore, the special name has been given to this one. Renal interstitium osmolarity. That is, just try to imagine cortico-medullary border, right? And then renal interstitium is the deeper one, going up to the end of the pyramid or the starting of the calyces, right? Now, renal interstitium osmolarity increases as we go from renal cortex to deeper in the medulla. That is, from the cortico-medullary border, when you go deeper and deeper, you will find increase in the renal osmolarity. That is, in the cortex, the osmolarity is 300 milliosmol, agreed. And just below that one border, again it increases, and from 300 to 400 to 500 and up to the level of hairpin loop of Henle, hairpin loop, it becomes a 1200 milliosmol. So renal osmolarity increases. This is from the cortical region when we go deeper. Osmolarity in the cortical interstitium is around 300 milliosmol, which becomes 1200 milliosmol in the hairpin band of the loop of Henle, and that is only in the loop of Henle of juxtamedullary nephron. Now try to remember, Cortical nephron does not involve in the concentration of the concentrated urine formation or diluted urine. It is only the juxtamedullary nephron whose say loop of Henle goes very deep, that is responsible for making concentrated or diluting urine. Increase in the osmolality is due to what? Why do we have it? Increase in the osmolality? Because increase in the osmolality is due to gradual increase in the solute concentration in the deeper medulla. Now, when we say solute concentration, we mean to say salt concentration, that is sodium chloride, potassium chloride, sodium sulfate, sodium bicarbonate, like that, right? And at the same time, these are the salts, but there is another solute, that is urea. Urea is responsible to provide nearly 50% of the total concentration. A huge quantity of urea is present in the renal interstitium. So, ladies and gentlemen, the ECF, that is the extracellular fluid in the deep renal medulla, is 4 times salty. Now, salty doesn't mean that it contains salt only; it contains urea as well. And then that of the cortex. The cortex, you can say osmolality in the deeper medulla is 4 times higher than cortical osmolality. And this is due to salinity gradient. I'm using the word salinity. That doesn't mean that only the salty part, right? Because it is maintained by a system known as counter current multiplier mechanism. Ladies and gentlemen, this is always a confusing part. Counter current multiplier system, counter current multiplier mechanism. That is responsible to maintain a osmotic gradient in the medullary region. Now, how does it work? Loop of Henle acts as a counter current multiplier. How it is a multiplier? How is a counter? What is the meaning by that? In the loop of Henle, the tubular fluid flows in opposite direction. First it goes downwards and then it goes upwards. So, this is the counter current. Going in the opposite direction means a counter current. Current is first downward current and then upward current. It is a multiplier because it adds NaCl, KCl from the thick segment into the medullary interstitium. Now just try to remember that is the thick segment of you may call it as a downward is a descending segment, is permeable to water but not to the solute, right? And then coming up, ascending limb of the loop only, thick segment is not permeable to water. Descending segment is permeable to water but not the ascending segment. What happens in the ascending segment? Now there are syn ports there which are responsible to cause the removal of sodium, potassium and chloride from the tubular fluid into the interstitium. That is, they add the salts in the interstitium, right? So interstitium becomes concentrated. It is again and again added, and therefore it is known as the multiplier, right? So it multiplies the salt concentration in the interstitium that is exclusively the thick segment. Exclusively the thick segment is responsible for this kind of the thing. Now this is the simple tubule, renal tubule. Here we find Bowman's capsule, proximal convoluted tubule. The fluid is flowing through. This is the thick segment of proximal convoluted tubule. This is a very big one. It must be up to this level, slightly wrong drawing. And then this is the loop of Henle, and this is the thick segment of ascending limb up to this level. A huge thick segment that is responsible to add sodium, potassium, and chloride actively by syn port here it is happening here now. See, you can see it. The sodium is coming here. Sodium, potassium, chloride, so they are taken out. I mean, this is the renal tubule we were talking about, and then come to here. Now, in the next, this is the cortical region. The blue color is cortical region, and remaining is the medullary region. What is happening here? Now, see, the tubular fluid in the proximal convex tubule is with a 300 milliosmol, right? When it descends down in the descending limb of the loop of Henle, two things are happening here. Descending limb of the loop of Henle is totally permeable to water, but not permeable to solid. Therefore, water is reabsorbed going to the interstitium, and fluid becomes gradually concentrated. When it descends down up to the level of loop of Henle, it becomes 1200 milliosmol, highly concentrated, and then it ascends up. During ascending up, it comes in the ascending limb of the loop of Henle, which is not permeable to water, but permeable to sodium, potassium, and chloride ions with the help of synthesis. That is the active transport of these substances into the interstitium. So interstitium concentration of these things increases, osmolarity also increases, right? And then this fluid goes to here it is absorbed under the. I mean, say these arrows, they show that there is an active reabsorption of sodium, potassium, and chlorides. Result is they are taken out of the fluid. Fluid becomes a diluted one, and it becomes a diluted part. It goes to the distal convoluted tubule. In the distal convoluted tubule, try to remember early part of the distal convoluted tubule and the later part. Early part is not permeable to water, right? But it is permeable to sodium, potassium, and chloride ions, especially chloride ions, sodium chloride ions. So they are absorbed under the influence of, your aldosterone. And then comes the later part. Later part becomes permeable to the water only in the presence of ADS, otherwise not. So this is how the fluid passes. Now try to remember here again, little bit applied part. In the proximal tubule, the exchange of the sodium is usually with the hydrogen ion as well as chloride ion. So chloride ions are reabsorbed passively as a result of osmotic pressure, and hydrogen ions are transported actively in exchange with the sodium. Hydrogen ion is produced as a result of dissociation of the bicarbonate, the carbonic acid. So here, whatever the amount of solute is reabsorbed, water is also reabsorbed. So it remains 300 milliosmol, the same milliosmol as it is in the extracellular fluid. Now, in this one, ladies and gentlemen, try to remember, in the proximal tubule, it is the carbonic acid which is responsible for the reaction between water and carbon dioxide. Carbonic acid can be inhibited by acetolamide or diamox. So this is when this is inhibited, lot of bicarbonate is excreted out, lot of water is excreted out. That is one of the diuretic. Then come to the ascending limb. That is the symport responsible for the absorption of sodium, potassium, and chloride. That is inhibited by Lasix or pyrazinamide. Now this is the ascending limb thick segment that contains a symport responsible for the absorption of sodium, potassium, and chloride ions, and thus the interstitium concentration increases, leading to, but this is not permeable to water, and therefore the fluid inside becomes diluted. Now you see 1200, 700, 400, 400, 200. That means more and more salt is removed, water is not removed. Now this is the early part of the distal contractibule, where the salts are removed but not water. So it becomes, I may say, diluted to somewhere 100 milliosmol. Right? Now try to remember it as the applied part. This is the part where your furosemide or Lasix works. It inhibits syn-port and therefore more urine is produced. Lasix is a diuretic which produces more urine by inhibiting the syn-port, and we call it as a loop diuretic. And then we come to this point here. It is an absorption of sodium chloride, and here the main substance is again a diuretic, which we give it to the people for edema and other substances. This is the part where 5 to 9% water is reabsorbed under the influence of ADS. ADS makes this is the collecting duct, make the cells permeable to water and urea. So urea comes out here, urea comes out here, urea comes out here, urea comes out here. So urea when coming out, there must be a decrease in the osmolarity, but that is further increased because the water is also going out. So try to remember this concentration gradient which is produced from here to here is partly from the salty and partly from the urea substances. Coming to the next one. Now here you will find it here. Something which is a multiplier system which we are going to discuss just now. Here again, just to revise, sodium chloride and water is reabsorbed here, it remains same. Here water is reabsorbed and therefore this becomes diluted. It reaches 600. This is only in the cortical region. When you drink a lot of water, then this leads to the precipitation of, ladies and gentlemen, come to this point. Now here, I just want to give you the idea. This is descending limb, this is ascending limb, and this is interstitium. Ladies and gentlemen, just imagine three things. Tubular fluid is present in the descending limb, tubular fluid is present in the ascending limb, and fluid is present in the interstitium. There is no change in the osmolarity. Imagine it. And then, at this time the fluid is stationary, no flowing. And then when we allow it to flow and come to this level, what is happening? Up to this level, 300. But when it reaches to here, it becomes 200. Why? Because what? Solute has been reabsorbed. Sodium, potassium, and chloride have been reabsorbed, right, actively. And therefore, the interstitium becomes concentrated and this fluid becomes diluted, right? And then come to the next one. Ladies and gentlemen, come over here. This is the interstitium which we had it 400 milliosmol. Now fluid is here. This is the descending, descending limb from the, this is the fluid from proximal contractibule, 300 milliosmol, coming down. It becomes 400. Further 400. Why? What has happened here? Because the descending limb is permeable to water but not to the solid. So water is taken out of here because here it is the more concentrated one. So concentration is high here, low here, water goes out and making this concentration equal. Right? Again here it is, again the same thing is happening here. So now see, here is what is happening. Because water has gone here to the 400. Once it comes over here, again it is reabsorbed. Potassium, sodium, and chlorides, they are reabsorbed. As a result of that one, this becomes a further concentrated. Added here, this is responsible. This is added in the interstitium, and therefore interstitium concentration osmolarity increases. Right? And this is the salt is added again and again, urea is added again and again, their concentration is multiplied, their level is multiplied, and therefore we call this one is as a multiplier system. Ladies and gentlemen, formation of the concentrated urine requires two main things: high level of ADH and high osmolality of the interstitium, medullary interstitium. Two things are important here. Now, ADH is capable to produce highly concentrated urine up to the level of 1200 milliosmol. And that is, I want to give you a little idea about it at this time. That is, in the event of a severe dehydration, which happens if we go to say outside field in the hot sun without drinking water, result is there is no urine formation, minimum urine formation, right? And this is happens because as a result of sweating, what body fluid is lost. Body fluid becomes concentrated. Is the osmotic pressure is high. High osmotic pressure causes two things here. That is we are going to talk about. Ability of the forming concentrated urine deeper in the presence of the osmotic gradient. Three major solutes contributes to high osmolality. They are sodium ions, chloride ions, and urea. Urea contributes nearly 50% of the osmotic gradient. Major portion of the urea diffuses from the lower CD, the lower collecting ducts. Two factors build the osmotic gradient: difference in the solute and water permeability, and counter current flow of the fluid through the U-shaped tube in the medulla. So counter current flow means flow in the opposite direction. That comes to, ladies and gentlemen, this is the new thing. I suppose you understand the meaning of counter current now. Counter current means repeated addition of the salts and urea in the interstitium again and again, and therefore this is maintained at this level. Then naturally a question comes to your mind: from the renal medulla, when the osmolality is high, renal cortex osmolality is low, why these solutes they don't diffuse to the other side? Because they are added again and again in this region, and therefore the concentration is always kept at that level. Another thing is that concentration is kept at that level by vasa recta. That is the counter current exchanger. This is something exchanges, so it makes the interstitium in a osmotic gradient. Now, ladies and gentlemen, what is this vasa recta? Vasa means vessel, blood vessels. Recta means a straight one. Now, these are the vasa recta are the counter current exchanger. How they are doing it? Vasa recta are the capillaries, straight capillaries arising from the efferent arterioles. Efferent arteriole of the juxtamedullary nephron. They are responsible for giving rise to the vasa recta. Now, vasa recta are straight vessels, then deep into the medullary interstitium, parallel to the descending limb of the loop of Henle. From the hairpin, it ascends parallel to the ascending limb and ultimately join the renal vein. So, vasa recta are the capillaries. When they arise in the descending limb, they supply oxygen and nutrient to the renal tissues, and in the ascending limb, they cause little bit excess. Now, flow in the blood flow in the vasa recta is quite slow and sluggish. Vasa recta is completely permeable. Now, these are the capillaries completely permeable to water and solutes. Now, what is happening here? When blood flows down in the descending limb of the vasa recta, deeper in the medulla, now just try to remember, they are the capillaries, they are permeable to water and solutes. Now, when it is descending down, what happens? It is descending from a lower osmolality to a higher osmolality. What is happening here? When it is descending down, it takes solutes and gives water to the hyperosmolar. Now, what is happening? What is happening in the blood? in the plasma or in the ECF of the blood. It contains solutes as well as water. Now, when this is descending down, the cells are permeable to water, but outside interstitium is hyperosmolar, so water goes out, and the solid inside becomes a concentrated. So by the time blood reaches to the loop of hairpin loop, it becomes a highly concentrated as interstitium. That is, it has taken lot of salts and it has lost lot of water. Now when it ascends up, now here water is lost, water is taken back and solids are lost. So it reaches back to that is in the beginning and end, the concentration becomes the same. When it descends down, it loses water and it gains salt. When it ascends up, it gains water and loses salt. So concentration becomes full. That means this is responsible to cause a mixing, some sort of exchanging. And therefore, this is known as exchanger. Ladies and gentlemen, hyperosmolar blood, while passing gradually decreasing osmolarity and there is no loss and no gain of the fluid or solute. This is the concentration of urine. How does it happen? This is quite an important one. I have put it in the smaller letters and a little bit details. Try to remember this one. This is not difficult to remember. Medullary osmotic gradients makes it possible to pass diluted or concentrated urine. This is done with the help of ADS. Ladies and gentlemen, I'll take your little bit time, say 2-3 minutes, not much. Now, medullary osmotic gradients, that is in the deeper medullitis 1200 milliosmol, in the superficial medullitis 300 milliosmol. This is the osmotic gradient. Now, this is responsible for making dilute or concentrated urine. And this is done with the help of ADS, antidiuretic hormone. Ladies and gentlemen, let us take late DCT, distal contractibule, and CD are impermeable to water. They are not permeable to water, they are impermeable to water. ADS from posterior pituitary acts on V2 receptors and induces water channel proteins that is known as aquaporin 2 in the luminal membrane. Luminal. So far I was using the word apical. Apical and luminal, they are the same. Towards the lumen, towards the apex of the cell. So apical or luminal border. Right? And helps in the absorption of water by the cells. This is the transcellular water reabsorption through the cells, transcellular. Under the influence of ADS, nearly 5 to 9% of the water is reabsorbed. 5 to 9% means 5 to 9% of glomerular filtrate, 5 to 9% of 180 liters. That is quite a huge amount. Nearly 15 to 20 liters of the water is reabsorbed under the ADS. Right? Normally, 1 ml per minute concentrated urine of 1010 to 1015 specific gravity is produced. Ladies and gentlemen, urine is expressed in terms of specific gravity. Normal distilled water is specific gravity is 1000. When the urine is a little bit more concentrated, it becomes 10,000, 1010 or little bit 10,050. So this is how it is compared. Now, in a state of dehydration, when there is a water deficit, as it happens in the severe loose motions and severe vomiting, and it happens in case of cholera disease, or in case of sweating, and the person working before the boilers or running a long race, right? That leads to dehydration. Dehydration of ECF. ECF osmolarity increases. When the water is lost from the body in dehydration, ECF osmolarity increases. That stimulates the osmoreceptor in the hypothalamus. Try to remember when we were talking about water balance in the beginning, in the first year beginning. That is, That is, so hypothalamus contains osmoreceptors which are stimulated by higher osmolarity of ECF, causing secretion of ADS and thirst sensation. So osmoreceptor produces two types of the two types of the changes. Number one is it causes secretion of ADS. Number two, it causes intense thirst. That is, when the thirst is there, we drink water. So replace water to correct dehydration. Another one is the ADS. ADS here will try to prevent the maximum water, try to prevent the loss of water at all. So ADS causes making reabsorption, increases water reabsorption from the DCT and CD, making urine highly concentrated. How it is done? It is done by aquaporin. Aquaporin is a water channel. So ADS causes causes receptor B2 receptor that induces this aquaporin protein in channels are induced in the luminal membrane or apical membrane. Water is reabsorbed. This is what is happening. How ADS produces increased reabsorption of water from the DCT and CD, making urine highly concentrated. Another thing which you might remember me in third year, fourth year, final year, or afterwards, minimum amount of the urine to excrete all the waste products from the body, the minimum amount is 500 ml. Normally we produce 1500 ml, but minimum amount is 500 ml. Less than 500 is said to be oliguria. In overhydration, which doesn't happen usually, but overhydration can happen as a result of excessive drinking of water or by our mistake. Sometimes we feel that, well, saline and glucose must be given intravenously. And we forget that how much is to be given. So that we are loading the body with the water. Overhydration as a result of drinking, in this one, there is no secretion of ADS. So whatever the water comes in the distal tubule and collecting ducts, that will be excreted out and no reabsorption of water in DCT and CDT, dilute urine is produced. So ladies and gentlemen, this is the story I wanted to give you. Thank you very much. God bless you. Thank you very much.
Thank you, sir.
Thank you, Mr. Thank you very much.